So one of the most exciting new activities here in the institution that I'm sure most of you are aware of is the development of cell therapies for the treatment of cancer. And David Miklos, our next guest from the Department of Medicine and the bone marrow transplant group, is in fact been one of the leaders of the group that has introduced CAR T cells into humans. David got his PhD and MD at my own alma mater at Yale and has been here for a very long time.
Yeah, we shared Yale. Thank you, Mark. Erin, that was a lovely talk. And big call out to Carrie Davis, row number 6, because I'm not sure why she's not giving this talk as the resident pediatrician. I thank Crystal Mackel for offering me up. Who knows what a CAR T is? Excellent. Okay, everyone's going to know by the time they leave. This is a therapy that impacts children through the therapy of ALL, acute lymphoblastic leukemia, and likewise mothers because of the problem of lymphoma. And finally, I'm going to say our best outcomes are octogenarian females. Seriously. So we're going to span all 3 generations. I have some conflicts. None of them are actually impacting my role in leadership or choices. Okay, so this is really the solution. That is, we have a big problem. Those diseases that are not doing well, that it would be relapsed acute lymphoblastic leukemia either before or after allogeneic transplants, or adults who have aggressive large cell lymphomas where the median survival of somebody who has failed 2 lines of chemotherapy is 6 months. Those were patients who were dying to get a new therapy, dying to be on clinical trial, and it's really a numbers problem also because just quickly showing the adult landscape, while we know about breast cancer, lung cancer, prostate cancer being the most common diseases, blood cancers are right up there at 150,000. And if you look at the breakdown of a pie chart, lymphoma and leukemia are the dominant problems. And if I just quickly say which of these types of cells express the target I'm going to talk about, CD19, everything in blue expresses. And while leukemia is a minor portion of what's going on here, it's a major proportion of what's happening with children. Okay, so I'm not going to try to make the case of, and this is a travel log, right? I'm not going to try to dwell on any one little thing too much. But really, this all happened in the last 3 years. In fact, 5 years ago, Ron Levy said, hey, that's Ron right there. He's the resident old man on the campus who does immunotherapy. And Ron said, hey, you want to go meet Steve Rosenberg? He's going to get a Nobel Prize someday. I said, absolutely. So we went to Steve's lab, and he told us all about this new therapy that this company called Kite wanted to buy and start marketing. And Crystal joined us in January of 2016, having been a branch director at the NCI in pediatrics and oncology, having 15 years plus experience of immunotherapies, and having done led a lot of what happened in childhood pediatrics. So what a perfect storm. This is actually the day we gave our first CAR T at Stanford. That is not very long ago, February 4th, 2016, and we've now given over 145. And this has changed the landscape. Everybody smiles when we talk about CAR Ts because 2 things. One is people are living and surviving, and yes, Erin, we need your help with some neurotoxicity issues. But the other thing is there's so much more to do. It is such an obvious area to move, and what we're going to tell you about today in lymphoma and leukemia is what needs to be applied to brain cancer, lung cancer, and so this concept of how do you target and hijack the patient's immune system and put it to work to cure cancer is what we're all about. So what's a CAR? It's a chimeric protein. It's a living therapy. We don't use the word genetic modifications. That's for GMO corn in Iowa. We say it's a living therapy where we take their patient's own cells and we supercharge them. We literally insert a gene that makes a molecule which is complex. And so if your own immune system is trying to, let's say, find, like Carrie Nadeau, an allergen being presented in an HLA context, and it's going to be restricted by what the specific HLA scaffolding molecules might be due to ethnicity, and then you have a very polymorphic T cell receptor. That T cell receptor is 1 cell out of 10 to the 10th. Okay, this is not likely to work. We're not likely to get the right combination, and we can't really vaccinate people when their cancer is killing them in 2 months. So what we've done instead is we've hijacked their lymphocytes, and we are expressing one molecule that will recognize a target antigen expressed on the surface of tumors. And what have we added to the armamentarium of cancer therapy? 4 things that never existed before. That is, we can take this cell, and it's, many times we talk about, oh, we painted the cell. We use military terms all the time, right? It's a smart bomb. This is an aircraft carrier, okay? This is literally dragging all those F-14s right to the scene of the crime, unloading its payload and going back, loading up with more and doing it again. It expands, it localizes the tumor, it uses cytotoxic killing, and it persists. And those four things were never accomplished with chemotherapy or even monoclonal antibody therapy. So this is my little fun thing. You can see in picture that the T cell is now expressing the CAR on its surface, and it has a kind of lock and key benefit that's going to recognize that it ran into the cell, but okay, let's say it was supposed to bind that antigen. And then it was going to, again, activate and make copies of itself, proliferate like 4 logs in 7 days. Become literally 50% of what's in the blood, and then release these cytokine killing molecules, and that tumor doesn't have a chance. Okay, so again, I said to you it was February of 2016, and this was our first version. This actually came after pediatrics. So homage to peds, this was their first therapy. They beat the adults to the concept, and this was done with Kara Davis. This is why I'm saying, why am I giving this talk? So Kara Davis has treated 10 plus patients in the leukemia pediatric system, and on a clinical trial that led to the FDA approval of Novartis' drug called MCHRI. I will speak to the adult drug, which we call Yescarta or Axicell in the non-trade name. And this experience now is what we started in February of 2016. So, I'm sorry, I jumped too fast. 3 different Kellers tells you 3 different parts of 3 different molecules put together in a chimera, and it binds like an antibody, it activates, and it stimulates for proliferation purposes. That's the car concept. And I use words like, oh, this is a Tesla, this is a Prius, because it's all about cars. And we put them in, they accelerate. They literally go from 2 million cells on infusion on the far left, and 7 days later, 4 logs more, where it's 50% of the cells, 50% of the T cells are now expressing this protein that's able to bind to a B lymphocyte expressing CD19. That expression and that expansion has side effects. Those side effects are things that we've learned how to manage. Everyone has met Emily White. She has allowed herself to be the poster child of CAR T out of University of Pennsylvania. And I think most of us have met Emily because, I'm going to jump ahead, she looks like this now, and she's 7 years out from her CAR, and the CAR still lives in her body. And it was Emily, the 4th patient, who taught us a lot of lessons because Carl June was able to see that the expansion of the CAR T cells caused fevers, right? That's fever curve. Incessant fevers. And then Carl says, I've seen this before. My kid has JR, juvenile rheumatoid arthritis, and she gets IL-6 blockade, tocilizumab, and he was the one who thought to do this. And it was a brilliant thought. And look what it did to the fever curve. And we've been learning more about how to control the CARs. I use words like we tap the brakes, we put it in low gear, we try to change these things. So much of what we've done over the last 3 years is simply build an owner's manual for the CAR that didn't come with one. And then also we're building toolkits on how to repair the CARs or how to understand and measure the CARs. 2 simple concepts. If you leave the room today and you say, I want to get a CAR because it sounds great, just know that there's 2 simple toxicities. One is called CRS, or Cytokine Release Syndrome, and it's the flu. If you had the flu, achy, body aches, muscle aches, joints, diarrhea, I have no appetite, the lights bother me. Okay, that wasn't the virus, that was the immune response to the virus. Now imagine you have a virus that's growing much, much faster. Patients have that fever, low blood pressure, and we call that Cytokine Release Syndrome. Everybody gets it, 90% of the adults get it, about 30% of the kids get it. The neurological events, we need your help, Aaron, is going to be the concept of, yes, sometimes people are delirious when they have high fevers, but it's also the effects of the cytokines and potentially some of the cells, especially the monocytes in the brain, and I'm sure it's more complicated than that, so we need that. And you saw that this curve goes up and the curve comes down. How they go up, how they come down, that's all the differences in the cars, and we bring out new car models every fall, so we're learning all the time. This is the excitement. I showed you curves earlier where everybody had median progression by 4 months and the 1-year survival was 12%. And instead what we have now is overall survivals that are out in 65% with lymphoma, ALL where everybody's responding to the therapy. Yes, we have things to fix, and we'll show you a couple of those things that Stanford's taking the lead on. But this plateau on the left and on the right is really impressive. And this is what gets us excited, because if you can do 50%, why can't we do 60%, 70%, 80%? It's even better when you meet the patients, so I won't be at speed dating, I'll be speed clinicking this afternoon. And so every Thursday, Friday, and now at this point we're seeing 5+ patients a week, and they are going to be very sick because they're literally going to, if they don't get this therapy, die in 4 to 5 months. So they come in very sick. And masses that are black in the abdomen over here after getting all the current therapies suddenly go away. And this patient's actually 2.5 years out. And again, 45% of the patients are actually durable responses. It's actually better than what we saw in the clinical trials, and we've done some real-world testing of this. So we have 2 drugs that are now available. And I use that word drugs kind of, I'm a little sheepish about that term because everybody's biotherapy is different. They use their own lymphocytes. We're not taking super donor lymphocytes yet, but that's coming. We're not able to predict exactly how everybody's response is going to work. We have to build those tools to judge the CAR. And now we have this medicine from Gilead, our neighbors up to the north, and Novartis, our neighbors in, well, I guess Switzerland. And they are very similar, as you can tell. The blue is the same binder. The green is the same activator. What's different? The stimulator. But they're very similar. Juno has one coming. So everything so far has been me too. What are the big problems? Well, we didn't know. So we started putting CARs in, and we had to kind of think, what do we want to understand? Well, how good are your T cells when we made the CAR? How good are the CAR T cells that come back to us? What was the process, and how did the manufacturing go? Did the T cells during manufacturing, do all the T cells express this CAR molecule? Do 10% of the T cells express it? Do they get to the tumor? Do they persist in the blood? We had none of the tools to test this, and so we've been generating these tools. All we knew was we got patients and we got tumor response on a PET scan, and everything else, build it yourself. So that's what Crystal Mackel, Kara Davis, myself, and a team of about 150 or 200 people now do in what's called the Cancer Cell Therapy Program. And so if nothing else, we're doing team science. We have to remind ourselves all the time that we're doing team science because it's hard. Yeah, like I don't have an ego. Okay, it's hard to always be working in teams and pulling in the same direction and making sure that everybody gets their time in the front of the room. We want to be able to think of a CAR as a bubblegum machine, right? Every one of those T cells in that tube after going through infection is different. It was a different T cell coming in, and it has a different expression of the CAR, and it has different potential, and how it measures in the blood, they're all different. So those colors of the rainbow matter, and we have to count them. And I'm just going to show one data slide. Okay, if I tell you that what's on the y-axis is the patient's amount of CAR T cells, this was patient 1 with the commercial, and this was patient 2 below. You could say they're very different. They're very different. The patient below was having seizures in the intensive care unit with all kinds of neurological toxicity. The patient on top, nothing happened, a little fever, no therapy, went home. They both had a complete response. So, and they're both alive. And they're both almost 2 years out. So I'm going to skip the busy slide. I'm going to show you something cool, though. That is, if I tell you that this is how the cells expand in some of our patients, that is not at all, because the cells are, he wants me back on the microphone, because the cells are going to the tumor. So you can see this man who has a calico cat situation with his spots. In the actual lymph node, when we start biopsying, and we've biopsied 150 lymph nodes now, the T cells are actually 50% or 30% CAR T by day 2. And even though there's nothing expanding in the blood, so you can see that we don't really care what's in the blood, we care what's in the lymph node. So we have to get there. Now, last concept, because I'm probably down to 2 minutes. This is the pediatric slide, so everybody pay attention. This was the pivotal study. It's called the Ileana trial. Again, Kara was a big contributor to this. Shannon Maud's the first author. And you can see at 28 days, the black line shows that the patients have a 90% plus response. The leukemia is in remission. But you can also see on that by 12 months, if they don't go to an allogeneic transplant, that the curve comes down. And what's the main problem? The expression of the leukemia cells for 19 goes away. Much like somebody who takes antibiotics all the time for a urinary tract infection becomes resistant to the antibiotic, well, so do these cells, and they don't express 19. So you can see that we're focused on 22. And so I'm going to tell you just in 2 slides about a double. So double makes more sense than a single, just like Mr. Bartender, make mine a double. And likewise, sequential might be important too. So we have an open trial for CAR22. And Kara, I'm going to tell you today, our diffuse large cell patient has almost a CR with a 10 centimeter mass at day 28. So we're the first to ever do that type of a CAR investigation with a 22 in lymphoma. And we're the first to actually do what I'm showing you here. Take patients who have negative 19 and give them a therapy that's going to bind to 20 or 22 and rescue them. So that concept's important, but what would be even better, I'm going to skip this slide, would be why don't we make a double? Let's make a CAR that binds both to 22 and to 19. Show that in the laboratory when the 19 is lost, that the cells still kill, and that those cells can persist. And then you have to test it in a patient. Now, how do you do this? Well, you have to make them yourself. I'm not Gilead, right? I'm not Novartis. Nobody brought me $12 billion. So instead, Crystal brought us Steve Feldman. Steve Feldman is in charge of manufacturing. He worked at the NCI for 10 plus years. He is like the guru who started the whole kite project, the NCI. And also, how about this? Your own car machine manufacturing at home, right? it actually looks like it, you just plug it in the wall and just everything works. Well, it doesn't work that easily, but this is a great break forward. And we have investments from Stanford at this point, over $20 million on GMP facilities, management, team science again. And it's complicated, but we have to take patient cells, manufacture them, and make them turn into cells. I can see I'm losing time. That's okay. As we look at people, they're all different. You got the idea. They're different. And you can study a trial now of 40 patients where we manufactured, and we saw these people who were dying, and we triaged and chose. And every Tuesday, we were able to collect a product, manufacture it, 10 days later, put it in, and be able to cure people. 100% of the ALL patients, 100% of the ALL patients have a complete response. And the current lymphoma looks just like the commercial product. So we're working on it. It's massive. It's great. I'm going to jump ahead, and I'm going to go all the way to the end because what I want to show you is this. It's not aspirin. And so it takes a team of people. It takes cooperation. It takes facilities. It takes an institutional commitment. And Stanford wasn't even on the map on this 5 years ago. Like, we didn't have a single patient getting a CAR therapy, and this has been developing for over 30 years. And I'm going to proudly tell you we're the top 5 now. So thank you, everybody.